All right, hopefully you have your notes. Um, these are the notes on the summary of events involved in contraction and relaxation of a skeletal muscle fiber. Those are the notes that you need for this exercise. I'm going to show you pictures in this next chapter. All right, so a little review here on this how muscles contract is we started off with the epimysium covering the muscle. Remember, it's a deep connective tissue which covers the muscle. It goes in and covers bundles of muscle fibers. A bundle is called a fascicle, muscle fascicle, and so there are 8 fibers here, and they're covered with the perimysium. So the epimysium goes in and covers bundles, and the bundle coverings is now called a different name, perimysium. And the perimysium goes in and covers individual muscle fibers as the endomysium. So we had epimysium covering the whole muscle, perimysium covering muscle fascicles, bundles of muscle fibers, and then each fiber is covered with the endomysium, which it points out down here also, covering a muscle fiber. And we have all these little myofibrils inside. So we saw that the plasma membrane of a muscle cell has a different name. It's called the sarcolemma. And the cytoplasm is called the sarcoplasm. And inside of the sarcoplasm are all these little myofibrils, groups of actin and myosin myofilaments overlapping each other. And this is a sarcomere from the Z line here. The other Z line is partially covered over here. That's one sarcomere that we'll look at in just a minute. But all these myofibrils are surrounded by the endoplasmic reticulum, which contains calcium, and now it's called the sarcoplasmic reticulum. And there's vessels that are storing calcium ions. And then the sarcolemma has tubules, which are lined with sarcolemma that go from one side of the muscle fiber to the other, one part of the cell to the other. They're called transverse tubules or T-tubules. And there it is down here, T-tubules. They go from one side to the other, and that's just a tubule made of the sarcolemma. The T-tubule is surrounded on either side by sarcoplasmic reticula. And since it goes sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum, that arrangement is called a triad. So again, we're kind of rehashing stuff. Well, here is a sarcomere of one of these myofibrils here. Here's one of the sarcomeres. Remember I told you there are millions of these in your muscle cells. And it goes from one Z line to the other Z line. That's one sarcomere, and it's labeled right here, sarcomere. You see that the sarcomere is made up of actin and myosin myofilaments. The actin is the thin red lines. It has thin filament. It's the actin. And myosin is the blue, the thick one. And you see that this sarcomere has an overlapping of actin and myosin myofilaments area, which is called the A band. The anisotropic is the dark band. And where there's just actin, here's this part of the sarcomere to the Z line, and then from the Z line, part of the next sarcomere just has actin. That's the I band, the isotropic band. It's the light band in between the striations. So the striations go like over here, dark, and there's a light area, and there's a dark area, and there's a light area. Those are the striations. And in between, where there's just myosin here in the middle, that's called the H zone. The H zone is in the middle of the sarcomere. This little M line is a protein that holds the myosin myofilaments kind of steady in the middle of the sarcomere. That's all it really does. Here is a real picture of a sarcomere from a Z line to Z line. See, this light area is just actin, just actin. The darker area, overlap of myosin and actin. You can see the H zone, where it's just myosin. And you see the M line, which is a little protein that holds the myosins together. All right, so now let's look at the actin and myosin again. Well, after this slide, it's showing you a sarcomere down here. And there's your triads. And when the calcium is leaking out, it's going to going to flood over top of these sarcomeres, and that's going to be important. It's going to have an effect on the sarcomeres. Looking at actin again, you see that actin is a double helix of the molecules actin. I told you, like, if you get a pearl necklace and you hold, latch it together and then you twist it, that's what this looks like. All the little actin molecules are spiraling like the beads of a pearl necklace would when you twist the necklace. Running lengthwise along this actin molecule is a complex called the troponin-tropomyosin complex. And what you see is here are two proteins. A long one called tropomyosin runs the length, and it's spaced at regular intervals along that tropomyosin is troponin. You see these little brown spheres here, spaced at regular intervals. And they're covering this yellow spot here. This yellow spot's an active site that myosin can bind to if it can see it, if it's exposed. Now, I showed you again how each myosin is surrounded by 6 actins. That's just my little model here. So when you look here, you're only seeing one actin, a myosin, and an actin like this from the side. One actin, myosin, one actin, and then a myosin, one actin, and myosin. But there's actually 6 surrounding each of these myosin molecules, which kind of explains why they look like this. The myosin molecule looks like a little golf club with 2 heads on it. There are actually 2 heads here. They call them cross bridges or heads. And there's a hinge, a very important part of that molecule, the hinge. It can bend, and you can see the little arrow there. The myosin molecules are bundled together. And so it makes them a thick filament, a thick myofilament, because there's a lot of them bundled together. You see that the heads are rotating around in a circle. These little lines here are the heads, the cross bridges, that are directed toward the actins, which are around them. So there's a lot of interaction between the actin and myosin when that sarcomere contracts. So let's look at a little bit of, starting at number 1 on your sheet. This is the impulse conduction pathway. Now, on your notes, it says a nerve impulse. Well, let's get this built first. Let's build this first. Let's get what it looks like. Here is a multipolar neuron. It has more than 2 extensions off the cell body. All the extensions are bringing impulses toward the cell body except one. The one that takes impulses away from the cell body is called the axon. So impulses are brought in toward the cell body by way of dendrites, and they leave by way of the axon. Now, the axon has extensions off of it. These are called telodendrites. Mine only has 3 telodendrites. They could have more than that. They could have 100. Or they could have as few as 3 like this. But each of these telodendrites goes to a little bulb, and you see that I've got 2 there. And here's my big one I blew up so we can see better. And that is suspended over a muscle fiber. So my motor unit here, my motor neuron, is activating or innervating or stimulating 3 muscle fibers. That's it. And we're going to look at this one down here. So we see that at the end of the telodendrite, there is a structure called the synaptic knob or the end bulb. And inside of that synaptic knob or end bulb are packets of neurotransmitter. These are called synaptic vesicles containing a neurotransmitter called acetylcholine. It's abbreviated ACH, acetylcholine. And I drew the molecule with a little box and a little circle for acetylcholine. It has 2 parts. And they're real little bitty in here. It's kind of hard to see them. The membrane right here of this synaptic knob that is facing the sarcolemma of the muscle cell, okay, is called the presynaptic membrane. Presynaptic membrane of the synaptic knob, because the space in between the 2 is called a synaptic cleft. That's a space. It's a physical space. They do not physically touch. There's a gel in this area, and this acetylcholine is going to have to diffuse across that synaptic cleft to get over here to the postsynaptic membrane, which is the sarcolemma. Well, you can see that it'll be released, and it's going to bind to receptor sites in an area called the motor end plate. That's where they're going to come across and bind. And if enough of them bind there, the impulse is going to be started up again. So let's look at another version of this same slide and follow an impulse. Okay, so number one, a nerve impulse, which is called a nerve action potential, that's what impulses are, leaves the neuron by way of the axon. There's the axon. The axon has several extensions called telodendrites, which deliver the impulse to the axon terminals or synaptic knobs. So that's that blue line is the wiggly impulse, and it gets down here and it depolarizes the membrane of this synaptic knob, which sets into effect a bunch of reactions we're not going to be concerned with right now. They do involve calcium, and what happens is the synaptic vesicles in here are going to be moved over to the presynaptic membrane surface. So let's keep reading. The impulse depolarizes the membrane of the synaptic knobs, okay, which causes the synaptic vesicles, so vesicles containing acetylcholine, the neurotransmitter molecule, to move to the presynaptic membrane of the synaptic knob. This membrane of the synaptic knob, which is above the synaptic cleft, fuse with the membrane and release acetylcholine into the synaptic cleft, and you see it's been released into the synaptic cleft. Acetylcholine diffuses across the synaptic cleft. That's going to be by diffusion. Like I said, remember, diffusion is the movement of ions or molecules from an area of greater percentage or concentration to an area of lesser. Well, there's a whole lot where it just got dumped out into the synaptic cleft, and there's none over here. So it's going to diffuse from where it's more abundant to where it's less abundant, and binds with receptors. There they are on the motor end plate of the postsynaptic membrane, or sarcolemma. So there they are. If enough acetylcholine binds, so that's a stipulation that we'll talk about when we get to the nervous system. If enough acetylcholine binds to the receptors, an action potential or impulse is created. It spreads over the surface of the sarcolemma, and then we're going to see where it goes after that. It goes down the T-tubules. So we got through number 1 and number 2. So we know that here's the sarcolemma here, there's a sarcoplasm in here, and here's our myofibrils inside with the sarcomeres. And you see that here's the T-tubules going all the way across, and the sarcoplasmic reticulum, which contain calcium. Now, as that impulse is going to go across the sarcolemma, go down the T-tubules also, because the T-tubules are also composed of sarcolemma, plasma membrane. And when it goes through there, it's going to depolarize the membranes of the sarcoplasmic reticulum. So let's look at a slide which I have a little drawing of the same events here, just a little easier to look at. There's a sarcolemma, see your triad there, sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum, and I have all the calcium inside of the sarcoplasmic reticula. And I've labeled this here for you to see what a triad is. So it's a little easier maybe to understand this concept of what we're going to be talking about. And again, there's what I have: sarcoplasmic reticulum, T-tubule, sarcoplasmic reticulum. So, next slide. The muscle action potential, the impulse that's traveling across the sarcolemma, depolarizes the membrane of the transverse tubules. See, it's going across the membrane, but it's also going down the T-tubules from one side to the other. And as it depolarizes the membrane of the T-tubule, it's going to depolarize the membranes of the sarcoplasmic reticula here. So let me read your number 3. The muscle action potential or impulse depolarizes the membrane of the transverse tubules, which in turn depolarizes the membrane of the sarcoplasmic reticulum. The sarcoplasmic reticulum store calcium ions. You see I got a lot of calciums in there that are used, are stored, and they will be used for muscle contraction. This depolarization of the sarcoplasmic reticulum results in a release of calcium ions from storage into the sarcoplasm, which contains the sarcomeres. Remember the sarcomeres? There's the sarcomeres that are in there. So let's look at a really, really big blow up. You see this little box here? That's basically what this is. You have an actin in the middle. They should have raised it up a little bit. There's your actin with your troponin tropomyosin complex there. Here's your myosin above and your myosin below. So it actually is that myosin, and this is the actin in the middle here. Now this is a resting sarcomere. This is before the calcium's been released. So let's look at that this first. you see that the troponin tropomyosin complex is covering these yellow spots, which are called active sites. They're being covered, and so in the olden days, they called these active sites, they called them myosin binding sites. So the myosin binding sites are covered by the troponin tropomyosin complex. So the myosin cannot bind to them. Now myosin is already broken down ATP to ADP and phosphate, so they're energized. There's energy stored in these cross bridges. And if they have the ability to attach to actin, then the sarcomere is going to shorten. So let's look at the next picture over step 1. Calcium ions bind to the troponin of the troponin tropomyosin complex. So they're going to bind to the troponin molecules, which causes the troponin tropomyosin complex to move deep into the groove of the actin double helix. So they're going to slide down. See, they were on the outside. Now they're sliding into the groove. Now look what's exposed, the myosin binding sites, so which they call the active sites, right? Thus exposing the myosin binding sites or active sites on the actin. Now if those are called active sites or myosin binding sites, guess what's going to bind to them? The myosin. And the myosins have the energy to swing up and bind and do something called a power stroke. So let's look at the next slide. Step 2 here, cross-bridge attachment. Since the myosin binding sites are open, they're going to bind. So let's look at number 5. It kind of rehashes the ATP. An enzyme named ATPase splits ATP into ADP and phosphate, and energy is released. The released energy activates or energizes the myosin cross bridges, these heads. See, it's been broken down, they're full of energy now. The myosin cross bridges, which can now bind to the exposed myosin binding sites or active sites on the actin, and they just binded. Okay, so they're binding. And apply force. This is called a power stroke. And so look over here at the picture, step 3 here. Causes the myosin cross bridges to move towards the H zone. That's in the middle of the sarcomere, right? This movement results in the sliding of the thin myofilaments past the thick myofilaments. Okay, so when that happens, the Z lines are going to be brought together. So number 6 on your notes, the sliding of the 2 filaments draws the Z lines toward each other. The sarcomere shortens and the muscle fibers contract and the muscle contracts. So that power stroke caused the actin to be pulled toward the middle of the sarcomere, toward the H zone. And that's going to continue until these myosins butt up against the Z lines. So let's look at the next slide. It repeats over again. ATP is going to bind, and they're going to release. Let's look at number 4. Then look at number 5. They're charged, and they will bind again and do the same thing over again. Bind, power stroke. And it keeps on doing that until the sarcomere is completely shortened. All right, now what I've got here on the next slide is a comparison of a relaxed muscle and a contracted muscle. And you see that the Z lines have been pulled closer together. Look at the distance here in the eye. It's hardly exists here. So this is maximum shortening as far as it can shorten. All the sarcomeres have shortened. Now, acetylcholine, let's go to this slide. Yeah, I got to correct this one over here. Acetylcholine is inactivated or broken down by acetylcholinesterase, S-A-C-H-E. So I will fix this for your notes. A-C-H-E. ASE is an enzyme ending, and that's going to break down acetylcholine. And you see what happens there? As a result, acetylcholine no longer has an effect at the neuromuscular junction. Now, this happens because there are no more action potentials are being produced, and so no more acetylcholine is being deposited in the cleft, and the enzyme that has been there the whole time now has the upper hand. Now it can break them all down and stop impulses from traveling across the sarcolemma to cause the sarcomeres to shorten. Now, Since there are no more action potentials are being produced, calcium ions are actively being transported back into the sarcoplasm by calsequestrin and calcium ATPase, using energy from the breakdown of ATP. Okay, so the whole process reverses. The low calcium concentration in the sarcoplasm permits the troponin-tropomyosin complex to reattach to the actin, so it's going to slide back. The calcium's pulled off of the troponin-tropomyosin complex, it slides back out and covers the myosin binding sites, and so myosin can't hold on to actin anymore, and the sarcomere lengthens. It returns to the relaxing state. As a result, the myosin binding sites or active sites become covered. The myosin cross bridges separate from actin. ADP is resynthesized to ATP, and the thin myofilaments return to the relaxed position, which is this state again. Here is contracted, and when the process reverts, it goes back to the relaxed state. So sarcomeres return to their resting lengths, the muscle fibers relax, and the muscles relax. Hope that helps you understand this process. Remember, get your sheet and go through this lecture another time. Got to get it down. I may ask you some questions on your test, so I'd be sure you understand it or ask questions.